Hi everyone, welcome to the part 2 of the lecture of the chapter Principles of Inheritance and Variation presented by Neetshala. Let's get started. Before we begin, don't forget to like, share and subscribe to the channel so that you do not miss out any notifications. Let's start with the chapter. In this particular lecture, we will be discussing mainly 6 topics. The first topic is going to be sex determination, second topic is going to be pedigree analysis, third is human genetics wherein we will be dealing with genetic disorders, fourth topic is going to be chromosomal theory of inheritance, fifth is going to be linkage and recombination, and the last is chromosomal mapping. So let's get started. The first topic is going to be sex determination. Now, sex determination was devised by a scientist known as Henking. Henking devised the method of sex determination. And how did he devise that? He was working on the chromosome of insects. So he was working on insect chromosomes and he could identify that only 50% of the insects, only 50% of insects had XX condition as the last chromosome and another 50% had only one X chromosome. That is, the other chromosome was absent in the case of males. And this was what he identified. And he named these X like structures as X bodies, and later on, the modern biologist renamed it as X chromosomes. Now, how does these X chromosomes help in determining the sex of an individual? That's what we're going to discuss here. Basically, we have 3 methods of sex determination based on the chromosomes. The first one is known as, the first one is known as male heterogamety. In male heterogamety, what is going to happen? The males are going to have 2 types of allosomes, X and Y, or it can be X and an absence of a chromosome. Both of these can result in male heterogamety. And if males are showing heterogamety, females should automatically show the same type of gametes, or we can say it is homogamety. So let's start with the examples of male heterogamety. The first one is humans. In humans, as you can see over here, the males are going to represent 44 plus XY condition, that is heterogametic condition, and mother is going to show XX condition, and that is known as homogametic condition. Now let us see another example. The example that you're seeing over here is that of Drosophila. Now Drosophila here can show XX condition in the case of females. This is females and this is male, and males can either show XY condition or it can show XO condition, and this is determined by a theory which is known as genic balance theory, which is beyond our syllabus. So we are not going to the details of genic balance theory. Let's come to the topic. The next method, the next example that we have over here is that of grasshopper. Grasshopper and many other insects are going to show the XXXO type of sex determination, where the female is going to represent XX condition and the males will have one X chromosome and another X chromosome, as you can see, is absent. This is known as XO condition, or we can say X0 condition. Now let's move on to the detailed explanation of the various sex chromosome and how is it helping to determine the sex of an individual. As you can see over here, this is the example of Drosophila, and in the example of Drosophila, as you can see over here, in the females there are 2 X chromosomes, and X chromosomes are longer as compared to the Y chromosome. So in the males there will be one long chromosome, that is known as the X chromosome, and there will be a short chromosome which is known as the Y chromosome. Now another example that we can take is insects. In insects, the female are going to carry 2 types of X chromosomes, whereas males will have only one set of X chromosome. As a result, what is going to happen? If the female gametes fuses with the X of the male, it results in a female. And if the male female gametes react with or fuse or fertilize with the sperms which carry no X chromosomes, in that particular case that results in the formation of a male. So this is known as XX, X naught or XO type of sex determination. So let's move forward. Next, we are going to discuss the sex determination in humans, which we have been discussing from earlier classes. Males can produce two types of sperms, one is X and another is Y. Now, if the X is fusing or fertilizing with X ovum, because ovum can only of one type, that is X type, it can result in a zygote which is XX type, and that can result in a female. And the Y sperm, if it is fusing with the X ovum, it will result in a male because it is XY zygote. Now let's move to the second type of sex determination, which is known as female heterogamety. Now female heterogamety is seen in the case of birds. Birds shows female heterogamety or the ZW-ZZ system, where the females are going to represent ZW type of sex determination, sex chromosomes, and males will produce only one type of chromosome, that is Z and Z. So we can say that in the case of birds, it is going to be male homogamety and females are going to represent heterogamety. Now let's look at the special type of sex determination, which is known as haplodiplondic sex determination. And the haplodiplondic sex determination is seen in honeybees. In honeybees, always remember that the females are going to be diploid and males are going to be haploid. Males are always haploid and females are always diploid. So as you can see over here, the diploid female always produces a haploid egg. And by which process can it produce that? It is meiosis. So as a result of meiosis, a diploid female can produce haploid eggs. Now if this haploid egg reacts with, or we can say fertilizes with, a sperm which is always a haploid produced by a haploid organism by the process of mitosis, then it results in the formation of a fertilized egg. Fertilized egg will result in a diploid female. Now there is another case where we have an egg. This egg does not fertilizes with a sperm. As a result, what is going to happen? There is a formation of an unfertilized egg. And this unfertilized egg can result in a haploid male. So we can say that the males have no fathers here, right? Because this male is directly formed from the female in the parental generation. So we can say that males have no father, but this organism, that is the mother of this organism, has a father. So we can say that the males do not have father, but they have grandfather, and such a method is known as parthenogenesis. Now let's move forward with a question related to sex determination. This is a very recently asked question in Neet 2019 Odisha paper. So let's do this question. Match the items of column 1 with column 2. First one is XXXO method of sex determination, and we had studied that XXXO method of sex determination is where females are going to be homogametic. So A should be matched with 4. Sorry, we can say XXXO method is female homogametic, and also we can match it with grasshopper, right? So two possibilities are here. So let us move to the second option, XXXY method of sex determination. XXXY method is female homogametic, you can say, and XXXO method we can put it with grasshopper. So let's put A is 3 and B is 4. Karyotype 45 is seen in Turner syndrome, so C should be 1. So we can eliminate option number 3. Eliminate option number 3 because that is saying C is 2. Now let us look at D now. D is ZW-ZZ, that is female heterogametic. So we can say that D is 2. And D is 2 coming only in one answer. So option number 4 is going to be the answer. Let's move on to a very important topic of this chapter, that is pedigree analysis. Now pedigree analysis is basically the family tree, and it helps us to predict whether an organism in any of the future generations shows a particular disease or a trait. So before we move to the pedigree questions, we have to understand the basic symbols in pedigree. As you can see over here, a square is going to represent a male, a circle is going to represent a female, Now, if we have this symbol, then that means the sex is unspecified, like when the child is died. In the case of stillbirth, in such cases, we can use sex unspecified, and this is the symbol for that. And for affected individuals, we will use the same symbols that we used before, but here there is an exception. You have to shade that symbols. And mating is represented by a single line. Consanguineous mating, that is mating between relatives or closely related organisms, that can be represented with the help of two dashes, as you can see over here. And now if we draw a chart, if you draw a chart connecting one generation and the next generation, you can see over here that the parents are written always above and children are always written below. And next one is parents with male child affected with a disease, as you can see over here. The male child over here is affected with a disease. And now if we write simply this simple and write some number inside that, that means that there are 5 unaffected offsprings. It can be males, it can be females also. So now let's move on to some examples for pedigree charts. The first one, as you can see over here, this is an autosomal dominant. This is going to be an autosomal dominant trait because the parents the parents are affected. Either one of the parents or both the parents are affected, and one male child is also affected, but here mother was also affected. That means we can say that it is not a sex-linked disorder, right? Both the parents are affected in the first generation, so we can say that it is not a sex-linked disorder. Rather, it is an autosomal disorder, and since the parental generation also has the disease, so we can say that it is autosomal dominant. The second one is known as autosomal recessive. Now, what happens in autosomal recessive? The parents are unaffected, whereas the offspring is affected. Parents are unaffected, whereas offsprings are affected, as you can see over here. Now, the third case is X-linked dominant. Now, what happens in X-linked dominant? As you can see over here in the first generation, that is the parental generation, the female is affected. Now, the female is going to transmit the disease to the male in the next generation. And in the F2 generation, this male in the F1 generation is going to give only to its daughters. That is female to male, male to female in the next generation. In the F3 generation, it is going to be given to the males. So this type of an inheritance is known as zigzag inheritance. As you can see over here, it is following a zigzag fashion. So it is known as zigzag inheritance. Also, it is known as X-linked inheritance. And since the parent is affected over here, as you can see over here, the parent is affected, so we can say it is X-linked dominant trait. And if the female parent is not affected, if the female parent is not affected and the disease is happening in a zigzag manner, then it is known as X-linked recessive disorder. Here, the female parent is going to act as a carrier. And some examples which we have in our syllabus for X-linked recessive, that is the most important case. X-linked recessive example is sickle cell anemia, sorry, hemophilia and color blindness. Hemophilia and color blindness are sex-linked recessive disorder. So now let's start with a question. What pattern of inheritance does this trait shows? As you can see over here, look at this individual. Where did he get the disease from? His parents are unaffected, right? His parents are unaffected, but the offspring got a disease. That means that this is going to be an autosomal recessive disease. Autosomal recessive disease. Why autosomal? Because here a female is affected and here a male is affected, so both the sexes are affected by this disease, and so it is going to be an autosomal recessive disease. Now here is the time for a self-analysis. We have 5 seconds for analyzing the first question. 1, 2, 3, 4 and 5. Let's see what is this particular pedigree. The answer is going to be autosomal dominant trait. Now try the second one. You have another 5 seconds. 5, 4, 3, 2, 1. and the answer for this is going to be autosomal recessive. Now, why did this become autosomal dominant? Because the first generation is affected, and here the parents are affected and the offspring is affected. Here also the parents are affected, the offspring is affected, so it is autosomal dominant trait. Whereas here the parents are not affected but the offspring is affected. Here also the offsprings are affected but the parents are not affected, so it is going to be autosomal recessive trait. Let's move forward. This is a picture from NCERT. NCERT examples for pedigrees. You have to understand that the first one is autosomal dominant, as you can see over here, and the second one is going to be autosomal recessive. It is going to be autosomal recessive. An example for autosomal recessive is sickle cell anemia, and example for autosomal dominant is myotonic dystrophy. These examples are important. Let's continue. Next, we're going to discuss a very important topic from which 100% guaranteed question will come for the NEET examination. That is genetic disorders. So let's get started with the topic Mendelian disorders. The genetic disorders is divided into two types. Let me do the classification here. Genetic disorders are divided into two classes. One is known as Mendelian disorders and another is known as chromosomal disorders. Chromosomal disorders. Mendelian disorders are those which obeys Mendelian principles that we studied in the previous class, like the complete dominance, the law of independent assortment, law of segregation. All these are obeyed in the case of Mendelian disorders. And the examples for Mendelian disorders are mentioned over here, as you can see. The first one is going to be hemophilia. Second is color blindness. Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. These are the examples for Mendelian disorders, and they are following the inheritance pattern that is seen in the case of Mendel's crosses. Now let us look at these diseases one by one. The first one is hemophilia. Now, what you need to know about every disease, we have to know whether it is autosomal recessive, we need to know whether it is autosomal dominant or sex-linked recessive or sex-linked dominant. This is the first important point. And second important point is the inheritance pattern of that particular disease, the symptoms of that disease. These are the three things that we are supposed to know. So the first thing here, you can see it is sex-linked recessive disease. Now, why is it a sex-linked recessive disease? Because the females in the parental generation are going to be carriers for the disease, and they are going to carry this disease to the male progeny in the next generation. That is what is given over here. This is your NCRT's portion. So if you haven't underlined anything, you can make a mark of these important points. Its transmission from unaffected carrier female, that is important, to some of the male progeny. That is what is happening in the case of hemophilia. Now here what is happening is that this particular disease is transmitted. Now this is a problem with the genes, right? So as a result, there will not be production of a single protein that is a part of a cascade of proteins involved in the blood coagulation. Now, as a result, what is going to happen? This person will not have a proper clotting mechanism, and due to which, what is going to happen? If there is a simple cut, this simple cut can result in non-stop bleeding, and can ultimately leads to death of the person even. The heterozygous female, that is the carrier for hemophilia, may transmit this disease to sons only, and that is why it is known as a sex-linked recessive disease. Now, can a female become hemophilic? It is extremely rare, because mother of such person should have to be at least a carrier and the father should be hemophilic. But this hemophilic father will be unviable in later stage of life, right? Most probably he will die before the age of 18. So we can say he is unviable for reproduction. So we can say that hemophilia is not usually reported in the case of females. It is usually reported in the case of males only. Now, the female pedigree of Queen Victoria shows a number of hemophilic descendants, as she was a carrier of the disease. So Queen Victoria was also a carrier for the disease. She was not affected, but she was a carrier for the disease, and that is why hemophilia have got a fancy name. It is also known as royal disease. royal disease. Now let us see a picture to understand this better. As you can see over here, this is of a healthy individual. In healthy individual, there is a normal blood vessel, and if there is a small hemorrhage that is happening, that is a simple cut, that can result in clotting after some time. But in the case of hemophilic patients, what is going to happen? In the case of hemophilic patients, the clotting factors are absent. So if a hemorrhage is happening, the blood is unable to clot. As a result, there will be extensive bleeding, and that can lead to death of the patient. Now here is a question which is asked in Neet 2018. A woman has X-linked condition in one of her X chromosomes. That means she is having a genotype of XXC, let us take. This chromosome can be inherited by. Now, there is a male, normal male we can consider, that is XY. Now, we have to keep one thing in mind, that every males are going to get the X chromosome from the mother. Now that X chromosome can be either this healthy X chromosome or it can be this diseased X chromosome. So we can say that sons can inherit. Now what about daughters? Daughters are going to get one X chromosome from the father every time, and one X chromosome she is getting from the mother. So that X chromosome can be either this or it can be this. So daughters also has a possibility for inheriting the character. Daughters also has a possibility for inheriting the character, and they are asking about inheritance, not expression of the characters. Now if the daughter is going to get this particular chromosome, there is another healthy X chromosome, so this is going to be recessive and this normal X is going to be dominant. So that X is going to be dominant over XC, so she will be a carrier and not a diseased person. But they are asking about inheritance, and inheritance of this X chromosome, this disease X chromosome, can be shown by both sons and daughters, so answer should be 3. Let's move to the second disease, that is sickle cell anemia. Sickle cell anemia is abbreviated as SCA. Now this is an autosome linked recessive trait. It's an autosome linked recessive trait that can be transmitted from the parents to the offsprings when both partners are carrier for a gene. That is heterozygous condition. So I'll take a blank slide to explain this better. Sickle cell anemia can exhibit 3 different conditions: HbA, 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 HbS, and HbS, HbS. HbA will result in normal individuals. Normal individual matlab they are having normal shaped RBCs. HbA, HbS will result in 50% normal and 50% abnormal RBCs, but still the person will be able to survive. But the last one is a fatal case, HbS, where both the chromosomes are affected. And when both the chromosomes are affected, complete 100% of the RBCs will be sickle shaped. Now let's come back to NCRT. Yes, out of the 3 possible genotypes, only heterozygous individuals for HS, that is HSHS, sorry, homozygous individuals, that is HSHS or HbS shows the disease phenotype. Heterozygous condition, they are going to be the carrier of the disease, and there is 50% probability of transmission of the mutant gene into the progeny, thus exhibiting the sickle cell trait. Now, how is this particular sickle cell anemia caused? Sickle cell anemia is caused by the substitution, this is important, the substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin. As you can see in this picture, as you can see, this is an NCRT picture. You can see over here that the 6th position, the 6th position में जो protein है, if it is a normal person, then it is going to be glutamic acid. But if it is an affected person, as you can see over here, the glutamic acid over here is substituted by valine. And when it is substituted by valine, what is going to happen? It results in sickle shaped RBCs and not the normal biconcave RBCs. Now, What is happening in the base substitution? The base substitution is GAG. GAG of the RNA is changing to GUG of the RNA base. It is the RNA sequence that is changing. So what is the corresponding sequence in DNA? In DNA it is going to be this. This is the corresponding change in DNA sequence. And what is the change in RNA sequence? RNA sequence is GAG is converted into GUG. And this particular mutant hemoglobin that has a very low oxygen tension and that undergoes polymerization under this particular very low oxygen tension, and as a result of which it is going to change its shape from the normal RBC to a biconcave disc. Okay, normal RBC that is having a biconcave disc like structure that is like this. From this it is going to change to a sickle shape that is like this. Let's see that better. This is a normal RBC. These are normal RBCs biconcave shape, but these are known as sickle cells. Sickle cells means they are going to be sickle shaped, so they can also causes blockages in blood vessels because of their sickle shape. Normal RBCs can flow easily, but these sickle shaped RBCs, they will block here and there. Now let's see the next disease, that is phenylketonuria. Now, phenylketonuria is an inborn error. It is an inborn error of metabolism, and it is also an autosomal recessive trait. So till now we have studied three diseases. First one was hemophilia. Hemophilia is sex-linked recessive. Second one was sickle cell anemia. Sickle cell anemia, as we know, it is a autosomal recessive, autosome linked recessive trait, or we can say it is autosomal recessive trait. And now we have the third disease, that is phenylketonuria. It is again an autosomal recessive disease. Majority of the disease that we have to study under autosomal is autosomal recessive. Only one example we have to study for autosomal dominant, that is myotonic dystrophy, that we have already studied in the pedigree portion. Now, what is happening in the case of phenylketonuria? In phenylketonuria, the persons that is affected lacks an enzyme that converts the amino acid phenylalanine into tyrosine. Now, हमारे फूड में ये दोनों अमीनो एसिड्स होते हैं, यानी ये दोनों प्रोटीन्स होते हैं और प्रोटीन्स में अमीनो एसिड्स होते हैं, फिनाइललानिन और टायरोसिन. लेकिन फिनाइललानिन हमारे बॉडी में स्टोर नहीं होता, वो कन्वर्ट करता है टायरोसिन में. लेकिन अफेक्टेड इंडिविजुअल्स में ये मैकेनिज्म पॉसिबल नहीं है. As a result, the phenylalanine will get converted into another compound that is known as phenylpyruvic acid or other derivatives of this. And these derivatives will get accumulated in the brain. And once they get accumulated in the brain, that can result in mental retardation. And it can also result in brick red colored urine. These persons are going to show brick red color urine. What is the reason? Because the kidney cannot absorb this substance. Phenylpyruvic acid and phenylalanine cannot be absorbed by the kidney. As a result, it is excreted out through the urine, and this urine is going to show a characteristic brick red color. So that is about phenylketonuria. Now let's move to the next disease, that is thalassemia. Now thalassemia is again an autosome linked recessive blood disease. It is again an autosome linked recessive blood disease, and you have to remember that here the length of the globin chain is affected. We have studied sickle cell anemia where the quality of the hemoglobin is affected. Here what is happening? The quantity is going to be affected, right? Quantity is going to be affected because the length of the chain is going to be different here. Now, we have two such globin chains. One is known as alpha chain and another is known as beta chain. And here the length of both the alpha and beta chains are going to be different in two types of thalassemia. In alpha thalassemia, the production of alpha globin chain is affected, whereas in beta thalassemia, the production of beta globin chain is going to be affected. Alpha thalassemia is controlled by two closely linked genes, that is HbA1 and HbA2, that is present on chromosome number 16, and it is observed due to mutation or deletion of one of the more of the four genes. Now, more genes affected, the less globin molecules are produced. As a result, the length of the chain is going to be reduced. What is happening in the case of beta thalassemia? In beta thalassemia is controlled by HBB gene that is present on chromosome number 11, and that occurs due to mutation of either one or both the genes. Why both the gene? Because it is diploid organism, so there will be two copies, right? So here it was 2 into 2 4 copies in the case of alpha thalassemia, and here two genes are going to be affected, that is HBB. Now we have studied this portion earlier. Thalassemia differs from sickle cell anemia because sickle cell anemia, we have seen that it is affecting the quality, so it is a qualitative disease, whereas thalassemia is a quantitative disease. So this is the person's chain that we can see over here. The chain that we can see over here has a different shape. That means the length of the globin chain here, if it is this much long, it is going to be like this much long. The length of the chain is going to different. As a result, what is going to happen? Proper carrying of oxygen does not happen, so that can result in anemia, and anemia is a characteristic of thalassemia. Certain type of anemia is a characteristic of thalassemia. Now let us see the next disease, that is color blindness. And color blindness means the person is unable to distinguish between the colors, and that is again going to be a sex-linked disease, sex-linked recessive disease, sex-linked recessive disease. And four conditions are there for color blindness. These are the four conditions. XY or XX is a normal condition, XCY is going to be a colorblind male, XCXC is going to be a colorblind female, and XCX is going to be a carrier female. So if you are carrying out a cross between XCX, that is a carrier female, and a normal male, let's see what is going to happen. The parents are XY male, that is normal male, and we have a female which is XCX, that is a carrier female. This is a carrier female. So when we are carrying out the cross, we can understand that there are four possibilities for the offsprings. The first possibility is XCX, right? X chromosome from the father and this affected X chromosome from the mother can result in XCX condition that results in carrier female. Now second is when the father gives the X chromosome and mother gives the normal X chromosome, that is XX, that can result in a normal female. Now, the next case is that the child will get Y chromosome from the father and X chromosome, that is the affected X chromosome, from the female, that can result in an affected male. So if we have a carrier female married to a normal male, then the possibility that a child will be affected is going to be 25%. And if he is getting a Y chromosome from his father and the unaffected X chromosome from his mother, then it is going to be a normal male. So there is 25% probability that the child is going to be colorblind. Now, if we get a question, what is the probability that a male child is affected? Then it is going to be 50% of the male child, because this is the complete male childs, right? These are the different possibilities for male child. In that, half is going to be affected. So if they are asking what is the probability that male childs are going to be affected, it is going to be 50%. And if they're asking what is the probability that a child is affected, it is going to be this 25%. Now let's move on to the chromosomal disorders. Now chromosomal disorders are those which can result due to absence or excess of abnormal arrangement of one or more chromosomes. Now, failure of segregation of the chromatids during cell division results in gain or loss of a chromosome, and that is known as aneuploidy. Examples for aneuploidy are Down syndrome, Turner syndrome, and Klinefelter syndrome, which we are going to study shortly. And similarly, we have another term which is related to aneuploidy, that is polyploidy. Polyploidy is seen in the case of plants, and we will study more about polyploidy in the chapter cell cycle and cell division. Now let's deal with Down's syndrome. Down's syndrome is a genetic disorder. It is due to the presence of an additional copy of the chromosome number 21, and that is known as trisomy of 21. Trisomy means, as you can see over here, there will be 3 X chromosomes, that is 3 autosomes at the 21st position. So that is known as trisomy of 21. This was discovered by a scientist named as Langdon Down. It was discovered by the scientist named Langdon Down in the year 1866, and that is why it is known as Down's syndrome. Now here the affected individuals is going to be short statured. He is not having much height. He is having a small round head. Palm is broad and a characteristic palm crease. Physical, psychomotor and mental development is retarded. So this is what is happening in the case of Down's syndrome. This is a picture from NCRT. Congenital heart disease, big and wrinkled tongue, broad flat face, flat back of the head. These are also characteristics of Down syndrome. Now let's come to Klinefelter syndrome. Klinefelter syndrome is due to the presence of an additional copy of X chromosome. So the 23rd trisomy, we can say. 23rd trisomy can result in Klinefelter syndrome. Here it can result in XXY condition. In XXY condition, he is going to get an additional copy of an X chromosome, and when he gets an additional copy, what is going to happen? He is going to have more feminine characters than masculine characters. However, due to the presence of this Y chromosome, he will show overall masculine development, लेकिन उसमें breast development होता है, that is known as gynecomastia, and also another features like poor beard growth, tendency to grow fewer hairs, breast development or gynecomastia, female type of pubic hair pattern, wide hips, small testicular size, long arms and legs. These are some of the characteristics which is affected with these individuals, and these individuals are going to be sterile because, as you can see over here, the testicles are not well developed. They are very poorly developed, and as a result of which they are going to be sterile. The last disease that we have to discuss over here is Turner syndrome. Now, what is Turner syndrome? Turner syndrome is a disorder which is caused due to the absence of one of the X chromosome. That is, it is a condition that is 45 plus X0. That means she is not having one chromosome. It is absence, or we can say nullisomy, absence of a chromosome. Such females are going to be sterile. They're going to be sterile as their ovaries are rudimentary. They have very rudimentary ovary, poorly developed ovaries, as you can see over here, and she's having poorly developed secondary sexual characteristics, and she can also have brown spots called as nevi all over the body. So these are some of the characteristics associated with Turner's syndrome. Now let's do some questions related to this. First question, this is a revision from the previous class. The question says F2 generation in a Mendelian cross showed that both genotypic and phenotypic ratios are same as 1:2:1. And where does this come in? It can come in codominance, it can come in incomplete dominance, right? So 1:2:1 ratio phenotypic ratio में भी होता है, genotypic ratio में भी होता है in codominance. Now let's discuss the second question. What is the second question? Which of the following conditions correctly describes the manner of determining the sex in the given examples? Correctly is asked here. Okay, so let's get started. Let's read the options one by one. First one, homozygous sex chromosomes produce males in Drosophila. That is, XX condition produces males in Drosophila. Is it correct? No. Homozygous sex chromosome, that is ZZ, is going to represent the female sex in birds. No, it is female heterogamety, not homogamety. XO type of sex chromosomes determine male sex in grasshopper. Yes, that is a correct statement, so that is going to be the answer. Fourth one, XO condition in humans as found in Turner syndrome determines female sex. It is found in Turner syndrome, that much portion is correct, but it is not determining the female sex, so that is wrong. So option number 3 is going to be the correct answer. Now let's do the next question. A normal visioned man whose father was colorblind. So let us draw a pedigree here. This is a pedigree based on the data which is given over here. What is given? We have a normal visioned man. This is a normal visioned man whose father was colorblind. Was married to a woman whose father was also colorblind. So she will be getting XC from the father and X from the mother. And since this person is normal, we can say that he's going to get Y chromosome from the father and X chromosome from his mother. So these are the genotypes of the two individuals. Now, if I carry out the Punnett square, I can understand that it is XCX. Another possibility is XX. Another possibility is XCY. And another possibility is going to be XY. So how many percentage of the child will be colorblind? It is going to be 25 percentage, or 1 by 4th. So it is going to be option number 1. Select the incorrect statement with respect to hemophilia. What is the incorrect statement? It is an recessive disease. Yes, it is a recessive disease, but it is not a dominant disease. So this is going to be the answer. If both the parents are carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? If both the parents are carriers, so let us take it as XHXH or XTXT, whatever you want. It is XTX with XTX because it is an autosomal disorder. It is not a sex-linked disorder. So let us take it to be XCH, or we can take it as autosomes AH, A and AHA. So if this AH is coming in contact with this AH, that can result in AHAH, that is going to be affected. AH and A will result in AHA, that is a carrier. A and AH, again AHA carrier. A and A is going to be affected. So this is the only affected individual. So we can say it is 25%. Next question is a question based on pedigree. Now here, if I am observing the fourth generation, the fourth generation individuals are affected, but their parents are not affected. So it is going to be recessive disease. It is going to be recessive disease, but since a female is also affected over here, it is going to be autosomal recessive. So option number 4 is going to be the answer. Now let's look at the next question. Which of the following mostly appropriates hemophilia, mostly describes hemophilia? डोमिनेंट लिंक डिसऑर्डर नहीं, वो तो रिसेसिव लिंक डिसऑर्डर होता है. Yes, but it is X-linked recessive gene, right? So this is also correct, but this is more correct. So this should be the answer. Now let's discuss the next question. What is the genetic disorder in which an individual has an overall masculine development, gynecomastia? On seeing this word itself, we can say the answer. It is Klinefelter's syndrome. Now this is a question for your DIY today. Pause the video and comment the answer in the comment section now. Let's move to the next question. A disease which is caused by autosomal primary non-disjunction. Autosomal, since it is autosomal, it cannot be Klinefelter syndrome, it cannot be Turner syndrome, and it is non-disjunction. Non-disjunction means it is going to be chromosome related and not sickle cell anemia. So it is going to be Down syndrome. In which genetic condition each cell in the affected person has 3 sex chromosome XXY? Again, it's very simple. It is Klinefelter syndrome. So this is a very repeated question. Look here, this is Neet 2019, and again there was a question on Klinefelter syndrome, and here we can see a question based on Klinefelter syndrome, just a little twist. So this portion on chromosomal disorders is very important. 2017 में भी question आया, 2019 में भी question आया. Now, another 2 DIYs for today. First question is you have to say the correct statement. Pick out the correct statements from the following, and the second one you have to say what is the probability that their grandchildren is going to be affected. They are going to be grandchildren, grandson is going to be affected. So these are the 2 questions for your DIY along with the previous question. So 3 questions are there for today's DIY. You have to comment the answers for these 3 questions in the comment box below, and the winners' names will be announced in the next video. And these were the winners of the last day's DIY contest. Praveen Kumar Ray has answered both the questions correctly. Congratulations, Praveen Ray. And Ani Ambili, Dr. Prince, and Swati Live to serve. All these persons have also answered the questions correctly, but please do answer it like this. because he have answered it very correctly. Two questions were there for the last DIY, and these persons have attended only one question each. So please attend both the questions in this particular manner. You have three questions today, so you can put 1, 2 and 3 and type the answers corresponding to that, and your names along with your answers will be commented in the next video, and the answers for the previous day's video DIY is announced in the Telegram channel in the PDF of this particular video lecture. You can get the PDF at the Telegram channel at Neetshala, and there will be also a quiz on the same topic at 9 p.m. today. So please be a part of the quiz, and with this we are concluding today's lecture. Thank you and have a great day ahead.